Hello, everyone. Before I begin, I would like to thank the organizers and the scientific committee to the opportunity to show you these preliminary results on the influence of cover crops on enzymatic activity on vineyards in a semi-arid climate from the SANCHOSTHIRST project. With establishment of any crop, the soil where it is developed undergoes variations, and it goes from relative dynamic closed system to a dynamic open system with agricultural use. And it happens because the crop exercise an extraction on the soil resources, and it could promote the loss of the soil health, understood in this case as the capacity of this kind of soils to generate food in the future, because we are talking about vineyards. In this context, a question arises to the work group, that is, if can cover crops be part of the solution to this problem, because cover crops will favor greater enzyme activity due to the presence of plant remains, due to the action of the rhizosphere of the cover crops, and the less disturbance of the soil. Also because the cover crops could increase organic matter content and promote low erosion rates, among other benefits. In this context, the GP Soil SANCHOSTHIRST project aims to determine if the use of cover crops can increase soil health in woody crops in a context of climate change. The consortium of this project is made up of this institution, the Universidad Autónoma de Madrid, IMIDRA, the institution that I belong, from the INRAE, Agroparistec, and Institut Agro de Rennes, the Silesian University, and the University of Sassari. This project is working on the influence of the use of cover crops, taking into account the biological properties, the physical properties, and the chemical. And the objective of this work is to determine if the use of cover crops improves enzymatic activity in vineyard soils. We studied in this work 4 locations of between the 10 that SANCHOSTHIRST project has in Spain. One of the locations was Bodegas Fariña from the town of Toro in province of Zamora, Aranda de Duero from the province of Burgos, Finca Experimental El Encín that belongs to IMIDRA, and Bodegas Robles that is in Córdoba. In each of these locations, vineyards with cover crops were selected with nervy tail vineyards plots also. 3 per plot, 3 composite samples were taken, 3 between wine vines and 3 between the lines of the wines, and from 2 depths, to 0 to 10 centimeters and from 10 to 30. The samples were refrigerated at the field, kept refrigerated in the way to the laboratory, sieved, and kept refrigerated until their analysis. We analyzed 5 enzymes activities, 4 of them of the principal main biogeochemical cycles, beta-glucosidase, urease, phosphatase, and L-sulfatase, and the 5th enzyme activity was the hydrogenase activity as an indicator of microbiological activity. This table shows the substrate that we used to analyze these enzymes with a reaction time and the end products that were determined through a microplate spectrophotometry. And in order to know if there was a significant statistical difference, we used the Kruskal-Wallis non-parametric test. Here, taking into account all the four locations at once, we could see that the use of cover crops increased all the enzyme activities in both legs. And in this table, summarize the mean values of each enzyme activities along with the standard deviation, separated by the depth. And it can be seen that the teal plots have less standard deviation in all the cases. And this will indicate that soil tillage will be reducing the diversity on soil. On the contrary, we could say that cover crops show a higher variability between the soil enzymes, possibly due to the presence of higher presence and higher diversity of the plants that appears in these soils with cover crops. Also, we studied each location independently to deduce the possible influence of the different parent materials that each location has. And in the case of Arán, in Aranda de Duero in Burgos, the cover crops increase beta-glucosidase and L-sulfatase between 0 to 10 centimeters. The use of cover crops increase almost all enzyme activities between 10 to 30, no phosphatase. And the phosphatase didn't show an increase in this case with the use of cover crops. In the case of La Finca Experimental El Encín in Madrid, the use of cover crops increased almost all enzyme activities, no phosphatase, between 0 to 10 centimeters, and beta-glucosidase, R-sulfatase, and urease were higher to between 10 to 30 centimeters. In this case, also the phosphatase didn't show an increase in the cover plot with cover crop. In the case of Fariña in Zamora province, the use of cover crop increased almost all enzymes, no dehydrogenase, between 0 to 10 centimeters, and urease was higher in cover crop plot between 10 to 30. In this case, the dehydrogenase activity didn't show any effect of the management. Finally, the Robles in Córdoba. In this case, the use of cover crops increased the dehydrogenase activity in the most superficial depth of the soil between 0 to 10 centimeters, and between 10 to 30 centimeters